Bismillahirrahmanirrahim. We'll continue with chapter 8. Now, transport across cell membranes. We talked about osmosis and how that's important. Now, this is about molecules other than water. Okay. So, there are two methods by which things move across cell membrane. One type of transport is called passive transport. And when I say move across the cell membrane, either from outside in or inside out, they're very similar processes. So one is called passive transport. The other one is called active transport. The term active implies that it requires energy in the form of ATP. ATP is the most common cache or energy, stored form of energy in the cell, as we have previously mentioned. So sometimes moving stuff across the membrane does not require the cell to expend energy, and that's called passive transport. When the cell does require energy to do the effort to move things, it's called active transport. Now, passive transport, okay, across concentration gradient, okay, passive transport occurs only, like that, emphasis, only if there is an existing concentration gradient there. If there is no concentration gradient there, passive transport ain't going to happen, right? So passive transport occurs only across an established concentration gradient via diffusion or osmosis, okay? And then there are two types of passive transport. So here's passive transport, say. In both of these instances, over here and here, the two types, okay, you can see there's lots of stuff on the outside compared to the inside, and they're going to both move in here. One type of passive transport is simple diffusion, okay? So if there's lots of oxygen on the outside compared to the inside of the cell, oxygen is just going to walk into the cell just by diffusion, passive transport. So it doesn't even have to knock at the door. Hello, may I come in? No, it's going to come in, right? So oxygen just comes in by diffusion. That's passive type. The second type is if the molecule is bigger than oxygen, for example. It's not just going to walk across the plasma membrane. It's just going to bulldoze across the plasma membrane. No, if it's a little bit bigger, but it wants to come in, and if there's established concentration gradient, it needs a little bit of help. That is going to come through the proper channels, the proper gates, just like the gates in the fence across the house. So in that case, that's called facilitated. Facilitated means to help along, right? So the cell helps along because it had established carriers over here, or the protein channels, which allow the process to occur. But in both of these situations, without a protein carrier channel, or with the protein channel, no energy is expended by the cell. That's passive transport. Now, as we have talked about before, active transport is when the cell is trying to get rid of something or trying to get something in against the concentration gradient. Things don't flow against the concentration gradient, right? So to do that, the cell has to spend energy to bring either stuff in against the concentration or to expel things out against the concentration gradient. So whenever the concentration gradient is not in the favor of the cell, it has to put in energy to do the work. Does that make sense? Okay. Now, passive transport. Example, solute is coming through plasma membrane. Passive transport in the form of facilitated diffusion. Here's a transport protein in the membrane which allows things to go across like that. So these are three examples, okay? Simple diffusion, okay? Channel-mediated facilitated diffusion, carrier-mediated facilitated diffusion. Now, this is the difference between, like, you know, A plus versus A plus 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 students. There are two types of facilitated diffusion channels. Okay, remember, facilitated diffusion is just like, okay, they're not like really doing work, right? They're just facilitating things. But there are two types of them facilitated. One of them is just like an open gate. It's like a secret open gate saying, hey, come over here. Okay? That's this channel. It's just it's like a tunnel across the... Right? So this is called a channel. Like a tunnel. Channel-mediated facilitated diffusion. The difference between the channel-mediated facilitated diffusion and a carrier-mediated facilitated diffusion is different. You can see the obvious difference. It's not a tunnel. What this thing does is, this is glucose molecules. It sits over there, and this protein changes the shape. Okay? And then allows it to come in. Do you understand? So, but it's still with the concentration gradient. Okay? Now, why are these two? Right? This is like an open gate, right? As I said. Man, if you're going to have a fence, are you going to have an open gate? Because you have no control over who comes and who doesn't come, right? So, but here you have a closed gate. Who controls the key to the gate, you think? The cell does. So the cell can open that channel or close that channel. When it wants something, it can open that channel for the glucose and close it when it doesn't want it. So the cell has more control over here. And there's no just pure based on concentration gradient here, right? You follow? So a cell when it wants to control stuff, even in the case of facilitated diffusion, it will bring that stuff using carrier-mediated facilitated diffusion compared to channel-mediated facilitated diffusion. Does that make sense? Okay. So here, look at this. This is a quiz. Is this a excuse me, channel or a carrier? Is this a channel with a long tunnel inside of it like this? The stuff just goes through based on the concentration gradient? Or is this a carrier which changes shape like that? It's a channel. See, this is a channel. Now you can see in this here, water, whatever these tiny thingies are, they're moving across there based on the concentration gradient. Okay? Whereas a carrier, see, it changes shape. It goes over here, it changes shape, it comes down. They're both forms of facilitated diffusion. So here's some example. This is example of a carrier-mediated facilitated diffusion on the left side over here. This over on the left side, those are glucose molecules. It's changing shape, okay? It's not a tunnel, right? That's a facilitated diffusion with a carrier. Okay, now over here is a channel. See that? That's a channel. Now the glucose is not going to come through. It's only going to come through over here. Now this guy goes straight across because it's a what? It's a channel. So that's a channel and that's a carrier medium. Does that make sense? So they're both facilitated diffusion. If there was lots of the same stuff inside the cell, neither of those things would come in. They only come in if there's a concentration gradient. So, not that we need more examples. These are some examples of carriers. See, they're changing shape. Okay. Now, the cell could just lock this carrier. See, it would change it so it's clocked, so nothing comes in. Okay. They only work if the cell wants them to work. Okay. I think we pretty much got that. Now to active transport. Okay. So the difference here again is concentration gradient. To move things across. Now look at here, obviously it's going diffusion, passive transport. Now here, if the cell wants this last two bit of whatever this is, okay, the cell wants it, okay, it's like really wants that, whatever green thingies are. It wants even those two things too. But there's a lot of them over here. They're not going to come, so it has to come through active transport using ATP energy. So here's an example. Active transport. See? So active transport requires energy. Now to get rid of these leftover hydrogen across here, see energy. You see that? Because there's lots of them over there. How is it going to happen? Okay, so look at this. They're going to go across with energy. Does that make sense? Okay. That's active transport. So this is just a summary of some of the things, okay? This is facilitated diffusion with the channel, simple diffusion, facilitated diffusion with the carrier. And these are just the aquaporins of water carriers, a special name for water channel. We talked about this famous sodium potassium ATPase pump that works against the concentration gradient to move sodium and potassium across the cell membrane using ATP energy. Now this channel is doubly smart, right? Because it does two things at one time, right? You follow? So there are many channels, okay? There are many channels that do two things at one time. That means they carry two complementary things at one time, okay? Big molecules, how do they get inside and outside the cell? Let's say if the cell really wants something that's out there that's pretty big. How do they come in and how do they leave? Okay, how does that happen, you wonder? Okay, they're not going to come through channels or carriers, okay? Because they're bigger than the channels and carriers. It's just like, oh! You know what I'm saying? So there have to be some other method. Okay, so before we go into that, last part, how did they come in? We're going to talk about the carriers and channels that do two things at the same time, okay? So we talked about the ATP pump. They can bring two things at the same time, like this way, right? Both of them are going down. But the ATP pump that we saw before with the sodium potassium, they're called antiport. One of them goes this way, the other one goes out. You follow? So if they bring two things in the same direction, that's called a symport. If they bring two things in the opposite direction, that's called the antiport. Okay? So this is an example of ATP pump. Okay, that's, doing that. And this is an example of a symport. We lost our opportunity to observe that one, but you'll see how it's going to bring two things in at the same time. See? Two things in at the same time. Now this one over here, you'll see two things, opposite direction. See that? Okay, so there are many different types of transporters like that. Okay. These are like hitchhikers. This is a sodium potassium ATPase pump. It's a little more bigger diagram, okay? Different. Alright. Now, talking about those big things, how do they come in? Okay? Those processes are given special name. They're called endocytosis, exocytosis. Exo means out, like exoskeleton. Okay? Endo means in, like endoscopy, inside looking, right? So endocytosis, eating inside, bringing in the cell. Exocytosis is taking things out of the cell. So when the cell wants to grab big things, it does endocytosis. That's this. See how the whole plasma membrane kind of like dips down and forms a pit like this. It forms a pit like this. And then it pinches off into this, and that's how vesicles are formed. See that? So the plasma membrane kind of dips down, makes a little dimple, and then it kind of pinches off and makes a vesicle. This process is called endocytosis. On the other hand, if the cell wants to take the garbage out, so to speak, watch this. If it wants to take the garbage out, it takes the vesicle, and the vesicle, look, it fuses with the membrane. It fuses with the membrane, and then it exudes and spits out the contents like this. So this is exocytosis. Endocytosis, exocytosis. In and out. Watch. Whoa, ho, 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 ho. That's endocytosis. And then this is exocytosis. Now watch what happens in these growth processes. They're going to form a vesicle here. A vesicle forms, right? And when it's leaving here, the vesicles, it comes and joins the membrane. Now look carefully. The vesicle's membrane is part of the plasma membrane. So the plasma membrane forms the vesicle, and the vesicle it becomes part of the plasma membrane. That's why we said the plasma membrane is fluid. It's able to do stuff like that. It's not like cement. It's able to pinch off, come in, and go out and make new. It's a very lively part of the cell. So this is endocytosis. and exocytosis. Here's another example of endo and exocytosis like this. Okay? Now, animals can do the same. So this is an amoeba, see? Amoeba is just go and eat other stuff, and it does that by just going in there and going across and pinching it. This we don't have a whole picture, but this guy's after this little thingy over here, okay? And then it's going to eat it. Eating, if it's very big stuff, if it's eating it, that's given a special. It's called phagocytosis, to eat, the cell eating process. Endocytosis, exocytosis, and phagocytosis. This is an amoeba, okay? And so here is phagocytosis. It's eating a bacteria. Whole bacteria it's going to eat. This is what our immune cells do, our white cells do. They eat bacteria that try to invade us. Okay, that's called phago. Phago is when eating. Okay, so endocytosis, exocytosis for the small stuff. It's like small, and phagocytosis is when taking the whole bacteria in. You get that? Here's an example of a bacterium. See, that the cell is going to take, and it's going to mix with the lysosome here. Watch. Remember the lysosome? It eats up bacteria. Now this is a higher magnification of how the bacteria is going to draw on it because of those microtubules and stuff like that. Remember now here's the lysosome moving in, and it's going to eat. Do you follow? Do you see how the vesicles are formed? Because of the cytoskeleton of the cell is dragging it in. All the inside, the filaments are dragging it in, and then moving that vesicles towards the lysosome, and the lysosome is moving towards the vesicle, and they're going to unite, and that's going to be the end of the bacteria. Yes? Okay, see, the bacteria is dead, gone, okay? So, that's it for transport of materials across the cell. Next time, inshallah, limitations of the cell side, we'll continue from there. Until then, Assalamu alaikum.